Good morning students. Today we are going to discuss about the RNA. So we have in the last two lectures we have already discussed about the DNA structure and function. So today we are going to continue the topic with the RNA. So you know that RNA stands for ribonucleic acid, and it is a nucleic acid which is found in the nucleus as well as the cytoplasm. So till now we have seen the DNA, they are normally present in the nucleus, but this RNA, they are present in the nucleus as well as the cytoplasm. So cytoplasm. So they are present both in the nucleus as well as cytoplasm, and there are two main type of RNA. RNA के दो प्रकार होते हैं. One may be the genetic RNA, and another will be the non-genetic RNA. Non-genetic. So we can say that the RNA may be of two types. It may be the genetic RNA and the non-genetic RNA. So the non-genetic RNA, they does not carry any type of information, or they are not carrying the inheritary material from one generation to the another generation. But the genetic RNA, they contain certain information related to the protein synthesis. So the genetic RNA acts as a genetic material for some virus. So in case of some virus, we can say that the RNA, they are acting as a genetic material. So here in case of this slide, we can see that the DNA here, in case of certain virus, DNA is acting as a genetic material, but in some case of in some viruses we can find out that RNA, they are the genetic material, and they are responsible for the transfer of heredity or the function of a cell, or the you can say the phages are controlled by this RNA. So if the virus or any type of organism which contain the RNA as a genetic material, then they used to carry certain information, and the non-genetic RNA function for the protein synthesis. For example, transfer RNA, we can say that, and the ribosomal RNA, they are the non-genetic RNA because they does not carry any type of information, but they are you can say that they are the non-genetic RNA, and they function in the protein synthesis. So they play a very important role in the protein synthesis, and these are said to be the non-genetic RNA. So here we can see the ribonucleic acid. This is the strand of the ribonucleic acid or RNA. So generally the RNA molecules is a single stranded structure, but sometimes it may be a single straight structure, or sometimes they may be variously folded, or sometimes they may be coiled up on itself. So these are the different type of the structure in which the RNA may be possible. Now coming up to the components of the RNA. So we can say that each nucleotide of RNA is composed of the three components. You know it. First one is the sugar. So it is a pentose sugar called the ribose sugar, and it is 5 carbon compound and has a pentagonal ring structure. So you know that ribose sugar, the sugar which is present in the RNA, they are said to be ribose sugar, and if it is present in the DNA, they are said to be deoxyribose. So here in case of RNA, the sugar which is present is said to be the ribose sugar, which is a pentose ring structure, pentagonal ring structure. This is nothing but the pentagonal ring structure. Next, it is again attached with the phosphate group. So it is derived from the again phosphoric acid, and it help in linking the nucleotides during the strand formation. It is normally the single stranded structure, And here in case of the nitrogen bases, we have seen the nitrogen bases are adenine, guanine, cytosine and uracil. So instead of thymine in case of DNA, we have seen there was adenine, guanine, cytosine and thymine in case of DNA. But here thymine is replaced by means of uracil. So you can say that these are the 4 nitrogen bases which is present in the RNA, and the purines may be adenine and the guanine. These are the 2 purines, and the pyrimidines they may be uracil and the cytosine. These 2 are the pyrimidines which is present inside the RNA. So the strand of the RNA is a long chain of nucleotide joined by the phosphodiester linkage, and you can say that the nitrogen bases may be 4. We know that that is adenine, guanine, uracil and cytosine. There are 3 different types of non-genetic RNA which is involved in the protein synthesis in both prokaryotes as well as eukaryotic cell. That is the mRNA, messenger RNA, ribosomal RNA and the transfer RNA. So here we can find out both the 3 pictures of the mRNA or the pictures of the RNA, and these 3 types of RNA, they are non-genetic RNA. So this is ribosomal RNA, this is transfer RNA and this is the messenger RNA. So this is a pic of a transfer RNA where you can find out the structure of the transfer RNA. It is normally clover leaf model. Next is this is the ribosomal RNA, structure of a ribosomal RNA. Which is consisting of 2 subunit. One will be the larger subunit, another will be the smaller subunit. So here you can see the structure of the RNA. This is a single stranded RNA, and here it is involved in the protein synthesis after the translation. So when you can say that in case of eukaryotic cell, it is produced on the DNA or produced by the DNA and by the process of transcription, and after the formation it comes into the cytoplasm and then associates with the ribosomes. So you can say that all these 3 type of the RNA, either the mRNA, rRNA or tRNA, all these 3 types are produced by the DNA. So these 3 types are the non-genetic RNA because they are produced by the DNA. So in this slide we can see all the 3 types of the RNA. This is the single stranded RNA, this is the subunit of the ribosomal RNA, and this is the transfer RNA. Now we are going to discuss the different type of RNAs. That is first one, that is messenger RNA. So how this messenger RNA are produced or mRNA are produced? So in case of transcription, a portion of DNA that is unzips and mRNA composes a strand which is complementary to the DNA template. So this is a double helical DNA strand. So unzipping of the strand takes place, and then after the transcription a pair of or a strangled single strand of the mRNA is prepared. This mRNA which is synthesized inside the nucleus, these all things which happens in the nucleus, this mRNA which carries the messages, it comes out in the cytoplasm, and here they attach on the ribosome and then the protein synthesis will takes place. So this is something about the mRNA. So during the transcription, the portion of the code called the introns are spliced out. So during the transcription, certain codes which are said to be the introns, these are the unnecessary codes and doesn't code for any type of information. So these are spliced out and they are said to be nonsense codons. This codons does not code for any type of amino acids. and that's why these codes are removed out, that is said to be repairing of the mRNA takes place, and then the protein is synthesized. So here we can find out there are 20 different type of amino acids which is present inside our body. Second type of RNA is the ribosomal RNA, and it is also said to be the rRNA, and it is produced by the DNA, and it is again a single stranded structure but which is variously coiled upon itself, and in the folded region it shows pairing between the complementary bases, and it is said that around 80% of the total ribosomes, 80% of the total RNA is of this type of RNA. So you can say that rRNA, 80% of the total RNA present in the cell. rRNAs, 80% of the RNAs they are present inside the cell. And these ribosomes are composed of the large subunit as well as the small subunit. Large subunit, that is consisting of 50S subunit, and small subunit 30S subunit, and each of which is made up of its own specific RNA molecules. So what is the main role of the RNA? So you can find out the picture. In this picture it is consisting of larger subunit as well as a smaller subunit. The main role, now coming up to the role, the main function is the rRNA provides proper binding for the mRNA. So it provides a particular site in which mRNA can easily bind to it. So it provides a proper binding site for the mRNA, and it also protects the protein molecules under certain construction work. So you can say that the main role is to provide the binding site for the mRNA. Now the third type of the RNA is the transfer RNA, and this transfer RNA molecule is a single stranded structure, but it shows regular pattern of folding structure of RNA, and which can be explained by the two model, that is hairpin model, or maybe the next one is the clover leaf model. So here we can find out the structure of the clover leaf model, and in this we can say that it is transfer RNA, it is the smallest of all the three types of RNA. तीनों प्रकार के जो RNA हमारे पास हैं, इन तीनों में सबसे small structure का होता है. It is consisting of 75 to 95 nucleotides. And transfer RNA, these are very essential component of translation, where their main function is transfer of amino acid. So it acts like a vehicle, and it attaches the amino acid and it brings to a proper site of ribosomes during the protein synthesis, and that's why they are said to be the transfer RNA, because it transfers the particular amino acid, it activates the amino acid and transfer to the ribosome site. So what is the main role? The role is it carries a specific type of amino acid at the CCA end. So this is a picture in which we can find out the amino acid. So at the top here we can say that these are the amino acid, and it carries a specific type of amino acid at the CCA end. This is the acceptor end or CCA end, which you can see end of the ribosome during the protein synthesis, and it places the required amino acid properly in the sequence, and that's why this is said to be, or you can say that it is one of the vehicle which takes the amino acid to a particular binding site for the protein synthesis. So we have seen today the types of RNA, that is transfer RNA, ribosomal RNA, and the mRNA. So we have already studied the DNA as well as the RNA. So in the next lecture we are going to see the next topic. Thank you for watching.